My name is Rick and Soni, and I'm a research trainee under the supervision of Dr. Hennick at UW-Madison. Today I'm excited to share with you the effects of hypoxia on the oxidative reductive status of porcine articular cartilage. Introduction. In vivo, the physiological level of oxygen in cartilage ranges anywhere from 3 to 10%. Our recent work with ORI, or oxidative redox imaging, measures the autofluorescence of electron carriers such as NADPH, which is prominent in glycolysis, and FAD, which is associated in oxidative phosphorylation. This is important for the current study because mechanical injury and osteoarthritis often results in metabolic dysfunction. Objective. Therefore, the objective of this study was to quantify the effect of oxygen concentration and zone on the oxidative reductive status of cartilage as indicated by ORI metrics. Methods. We had 11 pigs aged 3.5 to 5 months old. We took out 6 mm plugs using a biopsy punch and bisected each of those plugs. 2 thin slices from each plug were taken for ORI. Methods continued. On the leftmost image, you will see a picture of the oxygen chamber. It has the tubes for the nitrogen gas perfusion, also an inlet for the nitrogen gas. There's a lid with a window and a hole for the oxygen sensor, and finally, there's the sample chamber, which holds the sample in place while ORI is being conducted. In the middle image is the oxygen probe that keeps track of the oxygen concentration in the chamber. There's also the source for the nitrogen gas, and finally there's the oxygen chamber itself that we looked at in the leftmost image. On the top right corner of the slide you will see a representative image of the NADPH channel, and on the bottom right you will see a representative image of the FAD channel. Results. On the left hand side are the results for the NADPH channel, and on the right hand side are the FAD. The plots are separated by 3 different zones, with superficial zone, the middle zone, and deep zone. The superphysiological condition is in cyan and physiological is in pink. The solid black lines indicate a significant difference because of oxygen tension in the NADPH plot. The dashed line indicate that the superficial zone was lower than the middle zone under both channels for the superphysiological condition. Discussion. The increase in NADPH intensity under physiological levels of oxygen indicates that there was a buildup of electron donors. This is most likely due to decreased oxidative phosphorylation activity, even though there was not a significant decrease in the FAD intensity. It is also possible that the chondrocytes increased the amount of NADPH produced under physiological levels of oxygen via other metabolic pathways such as the pentose phosphate pathway. This study is clinically relevant because mechanical overload alters chondrocyte metabolism. This study suggests that superphysiological levels of oxygen may actually worsen this metabolic imbalance caused from the mechanical overload. Conversely, it also suggests that the physiological levels of oxygen may actually serve as a buffer or an added layer of protection from this metabolic dysfunction. Thank you for listening. If you have any questions or comments, please direct them to our Twitter account.